So my name is Bryn Gertner, and I'm a graduate student at the University of Oregon. And I'm part of the graduate student Greaves, and we were the ones that put this together. But I did want to say that this wouldn't have been possible without the generous financial support of various organizations inside and outside of the university, so I wanted to take a moment and thank everyone. So the College of Arts and Science, the ASUO, SEEB, the Oregon Humanities Center, and the Bowerman Advised Fund all helped make this possible. So thank you for letting that happen. So why are we doing this seminar series? This is a screenshot from a website of a private company. For the bargain price of $199, you can send them a little bit of your DNA, and they will send you a bunch of statistical information about your health, what diseases you may or may not get as you age, and a little bit about your ancestry, so you know a little bit about your heritage. So how do they do that? And how reliable is this information? If I send them my DNA and they say I have a 75% chance of developing male pattern baldness, do I believe them? And how can this information be misrepresented? So I do want to walk through one little case study of one way that some of this information could be misrepresented from a paper that was published a couple months ago. So the title of this paper is Friendships Moderate an Association Between a Dopamine Gene Variant and a Political Ideology. So scientists find this really interesting because what this is actually saying is there's a genetic predisposition to your political ideology, but it's moderated by the environment. So it's both genes and environments that help predict whether you're going to be liberal or conservative. And if we explore that a little bit more, what they find is that if you have one version of a gene, then the number of friends you have will help predict whether you're conservative on the low end or liberal on the high end. If you have a different version of this same gene, then the number of friends you have doesn't have any relationship with how liberal or conservative you're going to be. So the authors are very careful in the discussion to say that we cannot declare that a gene was found for anything, and actually that the really interesting thing is that there's a gene and environment interaction for political ideology. So again, it's not what your genes are, and it's not just your environment, it's a little bit of both. So how did the news cover this one? Here's the original title. Here's what a science website said. Now, we've got the headline, which was awfully misleading. They did a good job of summarizing the research, and they did say, oh, okay, well, it's not totally your genes, but everyone wants to put a liberal gene in the headline, I think. Our friends at Fox News said, you might not be able to read this, don't hold liberals responsible for their opinion, they can't help themselves. Okay, so that's the news. What did the blogosphere do? Wonkette was actually a little bit nicer. They covered the environmental side of things and said that being liberal means that you are an awesome person with many sexy friends. So that's nice. So the point of this seminar series is not to lambast media or anything like that. What really happens is that we have an original finding that gets filtered through these different layers all the way into, now we're in the internet area, we've got everyone's opinion everywhere, and information gets lost along the way. And so the goal of this seminar series is to provide the attendees with tools to interpret the information at this level or at this level, and not having to rely on outside writers or opinionators or whatever to do it for you. And in that way we can figure out not only what our genetic information is telling us about ourselves, but also what it can't tell about ourselves. But where do we learn this information? Who's going to teach us that? Well, this is the importance of scientific outreach. We're in this genomic era, and Bill's actually going to define that for you, so I don't have to do it. The point is that a well-informed populace can engage in meaningful debate about what we do with genetic information as a society. And so, as scientists, it's our job to actually do the teaching. And that's what brings us to this lecture series. So over the course of this lecture series, we're hoping to provide you with a handful of people who can give you the tools to interpret all of this information for yourself, and also to get you thinking about what issues we're going to have to start addressing. So on May 3rd, the Oregon Humanities Center is helping us bring Dr. Lee Silver from Princeton University, and he's going to talk to us about the personal genome revolution, focused specifically on health issues and on how finding out genetic information about yourself might influence how you choose to live your life and things like that. And then on May 23rd, Dr. Carlos Bustamante will come and tell us about how genetic information tells us about human ancestry and various aspects of human history that were previously unknowable to us. And today, Bill Cresko is going to talk to us about what genomics means for our past, present, and future. Bill Cresko did his undergrad at University of Pennsylvania and got his PhD at Clark University, my apologies. He joined the University of Oregon faculty in 2005. Since then, he's given over 100 talks and published 40 scientific papers addressing fundamental questions in biology. In particular, how do populations evolve, and what is the genetic basis to these changes? And he addresses these questions using the threespine stickleback, which, when found in the ocean, has a whole bunch of bony armor plates on its side. And then as we find population in the lake, we find that they repeatedly lose that bony armor plating. So he's not actually going to tell you about any of that research today, but he is going to tell you about the genomic tools that have been developed in his lab to address these questions, and also to address questions that are more pertinent to the rest of us about issues in human health and human history. And then he's also going to talk about how he's using the same type of technology that's allowing us to change the way that we think about certain diseases, including cancers. So with that, I give you Bill Cresko.
Okay, thank you very much, Bryn, for that great introduction to the series. And first off, I'd like to thank all of you for coming out on a beautiful evening tonight. It's a testament to the University of Oregon and to the community that people are so interested in scientific subjects like this, and I really hope you'll be able to make it to the next two seminars. One of the defining aspects of a great university is the production of knowledge and the dissemination of that knowledge. And oftentimes that dissemination is through teaching in the classroom, but one of the things that Grebes is trying to do, and other graduate students groups on campuses, try to do it through these sort of outreach talks. I'm really pleased to actually be a part of it, so great job, guys. Okay, so this is what I'm going to tell you a little bit about tonight. So my goal here is to tell you about something about the genomics revolution. Obviously not all of it, there's too much to cover. And what I'm going to do is I'm going to tell you a little bit about some background and what the genomics revolution means for how we think about our history, our past, what we actually think about our present, and how we're actually thinking about our health, and what it might mean for our future. And I think this future part is something that Lee Silver, for example, will talk about quite a bit, because I think that what's going to happen in the near future with the genomics revolution, whether we like it or not, it's going to change the way we see ourselves and the world. And how do we actually see ourselves? Well, when we look around this room, we see both an amazing diversity in aspects of height and shape and color and hair and phenotypes. So each one of us is unique, but at the same time, each one of us has some strong similarities. So what are those things that make us unique, and what are those things that actually make us similar? Well, partly, as Bryn said, it's our experiences, our environment, but also it's our genes. And what do we mean by that? So what I'm going to do tonight is I'm going to tell you a little bit about how our genes and our environment interact, not only to make the variation that we see in ourselves, but also variation in things, for example, like common diseases. So many common diseases that afflict millions of people around the world. For example, like osteoporosis or the decreasing bone density have both familial or genetic factors to them, but they also have environmental factors such as diet or disease. So how do those things actually interact to produce not just the disease, but the probability of developing that disease? So if you think about many diseases such as cancer or heart disease, all of those have a combination of environment and genetic components to them. And the genomics revolution is really informing the way that we actually think about the genomic risk factor side of things. So I won't talk about the environmental side of things. My goal tonight is to actually step through a little bit of a basic primer, a background on what genetics is and how genomics builds off of genetics, and then leads us to how genomics is revolutionizing the way that we're actually thinking about ourselves. So what I have listed up here, show up here, is a number of headlines or front pages from two of the most prominent scientific journals, Science and Nature. And you can see over the years, there's been a number of covers that had to do with the production of different genomes from, for example, Drosophila melanogaster, the fruit fly, the chimpanzee, mouse, sea urchin, and in particular, the human genome. So some of you in this room will actually remember this right here. This is a picture of Bill Clinton and Francis Collins and Craig Ventnor. Francis Collins there and Craig Ventnor. So this was the announcement of the finishing of the human genome. And this happened in late 2000. The human genome wasn't quite finished, but basically we called it done. We moved the yard line a little bit. Basically, that finishing of the human genome moved us into the genomics era. And I'm going to define genomics in a second, but I'll just give you a little bit of foreshadowing here. But basically, the genomics era is hallmarked by the complete genomic information of an individual. For example, it ends up that one of the genomes that we sequenced was of Craig Ventnor himself. So having complete genetic information from this one individual. So we'll talk about what that actually means in a second. So here's the outline, or my goal for tonight, is to get through a few major concepts, relate those to genomics, and then actually hit a few vignettes, not go into anything in great detail, but basically some little vignettes about what genomics is telling us about our past. I'm going to tell you one of the big discoveries out of biology, the fact that we have what we can call a conserved genetic or genomic toolkit, and that'll make sense in a second. I'm also going to tell you a little bit about Mitochondrial Eve and Neanderthal genes. This is another exciting thing that's come out of genomic research in the last few years. Then I'll tell you a little bit about research that's happening around the world that's changing the way we think about ourselves as individuals, and related to research that's happening right here at the University of Oregon. So one of the other things that defines a great university is the production of knowledge, and I'll tell you a little bit about some work that's happening in a laboratory here. And then I'll tell you about what I think genomics really means for our future. So I'll tell you a little bit about work that I'm doing in collaboration with another laboratory at the University of Oregon studying individual genomes of cells. And then I'll finish up with the doctor will see your genome now, and I'll tell you what that means in a second and how that actually relates to the. So what is genetics? Let's just start off at that level right there. So what do we mean by genetics? Well, at the most basic level, when we think about genetics, we can think about variation in a human population. And this is just a picture right here of what at that time was Connecticut College and is now the University of Connecticut, of all of the men lined up in terms of height. And this basically makes a distribution of height in that population. So some of that distribution is determined by the environment, but a good proportion of that is actually determined by the genes. And so what happens is, what we can do is we can actually take and relate the average height of parents, for example, to the average height of the offspring, and that relationship can actually describe the proportion of the variation in that population that's actually due to the genes, the genetic variation that's actually in that population. This is actually one of the first figures that was produced by a man named Francis Galton in the late 1800s to represent this idea of heritability or inheritance from parents to offspring. I'm just going to see if I can turn off this light up front here. Do you know how to turn this? Do you mind grabbing that for me? Thanks. In much of the 1900s, work on a number of model organisms such as fruit flies or mice, for example, and a lot of it started by a number of researchers in the early 1900s, such as T.H. Morgan, represented right here, identified the idea of genes. So we can actually identify the factors that are segregating in pedigrees or familial relationships. And these genes have variants that will produce, for example, a red eye or a white eye, and those are termed alleles. So I'm not going to try to hit you with too many terms tonight, but when I hit necessary terms, I'm going to try to define them for you, because we use these terms as biologists not to be confusing, but because they actually make it easier for us to describe things. So mutations can actually happen in genes that will, for example, change a normal brown mouse into a white mouse, and those mutations are the change of one allele form to another, one form of a gene to another. And those can be inherited through families, and by doing so, we can get at or understand the mapping of genetic factors to phenotypes. And for most of the early part of the 1900s, the 20th century, the genetic factors themselves, what the molecules were, was not understood. But the genes themselves as factors could be studied. And then along came the modern era of molecular genetics, where basically due to the work of Rosalind Franklin and James Watson and Francis Crick in 1953, they identified the molecular basis of genetics, where we know that the heritable material in which genes reside is DNA. And that DNA material is inherited from parents to offspring just in the way as you would expect it to be if that was the material that was actually coding genes and variants of those genes or alleles. And since 1953, there's been a huge explosion in molecular genetics, and University of Oregon has played a huge role in that, having the first Institute of Molecular Biology established here in the 1950s. And part of the reason for that, and I'm not going to go through this figure on the right here, I just want to show you that we know a lot now about molecular genetics. What I want to do is just talk about what's called the central dogma of molecular genetics. How does DNA actually get the information in DNA get moved from the molecule into the variation that we started talking about when we look around this room? Well, the main idea behind this central dogma of molecular genetics is this, that DNA is a double-stranded molecule that's a helix, and this can be actually opened up. And when it's opened up, this long-term storage of genetic information can then be what's called transcribed into another form of molecule, nucleic acid, called RNA. And this RNA has a special name called messenger RNA. And it's that messenger RNA, from DNA to messenger RNA, that then gets moved to another part of the cell, and one more step happens. That messenger RNA then gets translated into a series of amino acids, this amino acid chain that's growing right there, and that amino acid chain then will become a protein. So you can think of DNA as the long-term storage, RNA as the intermediary, and the protein as the outward, the molecules that do a lot of the work in our body. So for example, the different aspects of our structural components of our skin or our hair or our fingernails, each one of those, many of those are made of protein. So DNA to RNA to protein. Cool. Okay, so we just got through the first few weeks of Biology 101. I know that was kind of quick, but that gets us to a starting point to think about, all right, so we know what genetics is, or we get the basics of genetics. What is genomics anyway? What's different about genomics than genetics? So when we say that there's a genomics revolution, what has happened over the last few years that makes us know things differently than we did a few years ago? Well, on the one hand, genetics or genomics is just genetics writ large. We're actually studying all the genes in organismal genomes simultaneously. So it's a natural extension of genetics. But I think it's actually a little bit more than that. Part of what genomics is understanding how these genes actually function in systems to make, for example, the final outward appearance of our body shape, our height. And as you'll see in a second, this is no simple task to understand that entire system because there are a lot of genes in a genome, and we'll talk about that in a second. Another aspect of genomics is that a large proportion of it is actually being driven by technological breakthroughs. So a little bit later on in the talk, I'll give you an example of a couple of those technological breakthroughs that are actually happening in genomics right now. And they've really been driven by increases in computational approaches and microscopy, and I'll tell you about that a little bit later on. And computers, actually, computer science. So if I had to actually think about genomics in two general areas, there's two main ways that I can divide this up. One is comparative genomics, where we go through and ask about how similar or how different are all of the genes in genomes of closely or distantly related organisms, and what does that tell us about their history? And then another aspect of comparative genomics that I won't talk about tonight, but is very exciting, is there's actually clues or hints in genomes that will actually tell us which regions of the genome were actually the objects of natural selection. Where does natural selection actually act in the genome to actually produce changes that are adaptive in organisms? Another aspect that we can talk about is functional genomics, where we ask, when we think about the central dogma of moving to DNA to RNA to protein, instead of doing that one gene at a time, what's the entire set of genes that are actually expressed, for example, in the developing tissue that's going to become a heart, or when a heart's actually damaged? What are the entire suites of genes that are actually transcribed and translated? So it's a study of gene activity and function, but also a good, an interesting component of functional genomics is you can actually change environmental conditions and ask how that changes what's going on in the genome. So I'll tell you a little bit about that tonight. Excellent. All right, so what I'm going to do is I'm going to switch gears right now and start talking about where genes are in the genome. And to do that, we have to think about where genes are in a cell. So all of us are made up of multiple cells. And I'm going to talk about animal cells, but similar things could be said about plant cells and fungal cells. And all of us are multicellular organisms. When we look at our bodies, we have millions and millions of cells. And when we look inside each cell, what we can actually see is that it's a very complex structure. But what we can focus in on are two main areas. One right here, which is the nucleus. And inside the nucleus of a cell, this is the outside of the cell, this is the inside of the cell, most of the genetic material or the genome of an organism, of an animal, lives and it resides in the nucleus of the cell. However, there's a little bit of DNA that's outside in the mitochondria, which is this structure right here. And we're going to come back and talk about mitochondria in a second. And so if that word's unfamiliar to you, it'll become a little bit more familiar later on. So where's the DNA reside? Where's the genome reside? In the case of animal cells, in two places, in the nucleus and a little bit that's outside of the nucleus. That's a little curious, but we'll talk about later on why we now know that DNA actually exists outside of the nucleus. So now we know that there are genes in the genome and that the genome exists largely in the nucleus and a little bit outside. How big are genomes anyway? What do we mean by that? Well, if we go back and think about the double-stranded molecule of DNA that I showed you, there were base pairs in that molecule, A's, G's, C's, and T's. And you're oftentimes seen it represented as just a series of them. But how many of those actually exist in a genome? The answer is a lot. There's huge numbers. So each one of you has 3 billion base pairs of DNA in your body. It's amazing to think about. But not only that, just look at your hand and look at the back of your hand. Every one of the cells that makes up your skin, your hair, each has 3 billion base pairs of DNA in it. It's amazing to actually think about how much there is in there. What is most of that DNA? Well, it ends up that most of that DNA is not actually genes that we talked about before that actually gets transcribed and translated into proteins. So these little pink dots right there represent the protein coding regions of the genome. And what I'm representing right here is human chromosome 1, and it by itself is 263 million base pairs long. So if we just focus in on a little region right there, most of that region of the genome is actually non-coding. About 99% of our genome doesn't get transcribed or translated. For a long time, we actually thought, or terms were used like junk DNA. As I'll show you in a second, it's not junk. We just didn't know what it did. We still don't know what it does. It's one of the big mysteries, or what I call borrow a term from physics, the dark matter of molecular genetics. What is going on in this non-coding region of the genome? So these genes right here exist sort of as islands in this large genomic sea. How many of them are there in an average genome? Well, it depends obviously on the different organisms that you actually look at. But in many organisms that are related to humans, there could be anywhere from 25,000 to 35,000 genes. So 25,000 to 35,000 genes, all doing things in every one of the cells of your body at different times, being transcribed and translated, and that's only 1% of your genome. It really is an amazing that these things exist in every one of your cells. So how are they actually exist in the cells? Well, what I showed you on the previous slide was human chromosome 1, right there. And human chromosome 1, like I said, was a very large fraction of the genome, 263 million base pairs. Now, I know it's kind of hard because we use these numbers like million and billion, but I just want to put this into context a little bit. So if each human genome has 3 billion base pairs, and they're represented in terms of 23 chromosome pairs, each one of these pairs of chromosomes is a condensed, in this figure right here, is a condensed amount of DNA that, for example, if you stretch this out in real space and just looked at it, it would be 8 centimeters long. It's a lot of DNA that actually gets condensed and wrapped up. That DNA in that chromosome as compared to this chromosome as compared to that chromosome exists in 23 chromosome pairs. One of these pairs you got from your mother and one you got from your father. If you're a male, you got one X chromosome from mom and a Y chromosome from dad. If you're a female, you got 2 X chromosomes. So to put this in context, there's 30 million books in the Library of Congress. So if each base pair was just representing one book, that would be 100 Libraries of Congress of books in every one of your cells. Not only that, every time your cells divide to make new cells, every time you slough off your skin, so the next time you clean up your dust in the corner of your house, just think to yourself how amazing this is, because most of that dust is probably your skin cells and a few other things. Every one of those skin cells at some point went through and basically replicated every one of those 100 Libraries of Congress of DNA and did so amazingly well. So if this was a scribe actually doing this, there would be only one mistake every billion base pairs. It's crazy how well the molecules actually work. So what does that mean? It seems like we should all be the same. Well, we're not. If you actually just sort of glance to your left and glance to your right, each one of us differs by about 30 million base pair positions. So each one of us is different by about 30 million. So we share most of those 3 billion, but we still differ by about 30 million base pair positions. Pretty cool. So how does DNA actually replicate? Well, DNA actually replicates through this process called semi-conservative DNA replication. And soon after the structure of DNA was actually discovered, the nature of replication was discovered by two researchers, Matt Messelson and Frank Stahl. Some of you may recognize Frank Stahl's name because he's been a faculty member at the University of Oregon for a number of years. And this has been called one of the most beautiful experiments in biology that they did. And it really is. Because before this point, people did not know how the double helix was actually unwound and duplicated. It could be that there was actually one copy here and another copy there. But basically, it gets unwound. And from each strand, the new duplicate is matched to the old strand on both strands. So basically what happens is each new molecule is half old and half new. It's interesting by itself, but think about the consequences of that. For all of the history of life in which DNA has been used as the molecule of inheritance, and this is the way that replication has been occurring, you can trace back through time two halves of a DNA molecule touching each other, and so on and so forth. So basically, everyone in this room could trace back through all of our ancestors DNA molecules that were physically touching one another back to the beginning of life on Earth about 3.8 billion years ago. It's pretty amazing to think that actually was happening. So how does this relate to evolution? And you'll see in a second why that matters. Well, that tracing back through time of the DNA molecules that are actually touching one another, going backwards further and further in time, gives us a history. So up until Darwin and Alfred Russel Wallace proposed their theory of evolution, and largely Charles Darwin put this together independently from 1830s and up until the 1850s, until Alfred Russel Wallace independently came up with the same idea years later, and then proposed this to Darwin unknowingly that Darwin already came up with this idea, and Darwin basically said, I had that idea a few years ago, and Wallace, to his credit, said, you're right, you did. Because if you look in Darwin's notebooks from when he was traveling around the world in the 1830s, he drew one of the first phylogenetic trees, if not the first phylogenetic tree. And what I mean by phylogenetic tree is an ancestor-descendant relationship of organisms with respect to one another. It was at that time that Darwin said to himself, and then later to the world, that all of us are descended from a common ancestor on the planet. And as such, at that time, he didn't know this, but we now know that the reason that we have similarities through the ancestral descendant relationship is that the DNA molecules were being passed on from parents to offspring over all those many thousands and millions and billions of years. So what has happened during that time is that in those years that led to the differences is that even though the DNA copying process that we just talked about is very efficient, even over that extended period of time, mistakes can happen, and those can be passed on from generation to generation. So those mistakes or the errors in the copying of the DNA, those scribes are very good, but every once in a while they make a mistake, that then becomes a mark or an indication of the history of that lineage, and then all subsequent ancestors actually carry that history. So what Darwin and Wallace came up with were two things. One was the evolutionary pattern part of evolution, which is that all organisms are related to one another via a pattern of ancestral descent, But then they also came up with a mechanism, which is that organisms that had these new variations that arose that actually could survive and reproduce to a greater extent would more likely pass along those variations to subsequent generations. So it's the mechanism as well as the pattern. So I'm not going to talk about the mechanism at all tonight, but the pattern as well too. Okay, so what we now know is that there's this great tree of life that exists. If we look out across the planet and we look at all the organisms that exist, Darwin was right. We can actually trace them all back to a common ancestor. And depending upon how similar the organisms are that we look at, for example, if we look at mammals, we can trace them back to a common ancestor that existed not that long, a number of years ago, but not nearly long ago, as if we looked at three major groupings here. And these groupings are called the eukarya, archaea, and bacteria. And I'm going to define these here in a second, just because we're going to come back to these in a little bit. So you are here in this tree. So this tree is not to scale at all. This tree right here of eukarya, this is basically all of the life on Earth. So most of the organisms that exist right here, at some point, DNA and thus genomes evolved way down here a long, long time ago. How long ago? Billions of years ago. It's an amazing long time period ago. And when we look at this tree, this clade right here in which we exist, this is the clade of organisms that have plants, fungi, animals, and some other organisms. But basically, this is most of the world that we think of as being sort of macroscopic plants, animals, and fungi. But in this region, right down here, the archaea and the bacteria make up everything else. So these are all the single cellular, what we might call microbes that exist out in the world in amazingly diverse places, from the tops of mountains to under glaciers. And they actually make up a huge amount of the diversity of life on Earth. And as you'll see a little bit later in my talk, even though they make up a huge amount of the diversity, we know relatively little about them. And even more importantly, these organisms have existed on the planet much, much longer than any of the organisms to which we're related in terms of the eukarya. So these are the eukaryote lineage, and this is the prokaryotic lineage. And prokaryotes have existed on the planet probably 2 billion years longer than the rest of the lineages that we think of plants, animals, and fungi that most of us normally think about. Okay? All right. So let's go back to this. So now we've got sort of the overall definition of genetics, genomes, and how it fits into a grand sweep of the history of life on Earth. And we now know that some of that history is actually written in the genomes. What I'd like to do now is start telling you about some of these stories that are written in the genome. And I apologize, I know that in the last 15 minutes or so that was very quick over a lot of biology, but what I really wanted to do was just to give you some of the basic ideas and terminology to know that what we can now do is go in and sequence genomes and start to tell some stories about our history. So I'm going to start telling you some of those stories. So let's first look at nuclear DNA that's in the genome right there, and let's compare it to this mitochondrial DNA. So as I said, most of the genome actually exists in the nucleus right there, but a little bit of DNA actually exists in the mitochondria. So this is this region of the cell that's outside the nucleus. Where did that DNA come from? It was a mystery for a number of years. But a scientist in the 1970s named Lynn Margulis proposed that maybe that DNA was actually from somewhere else. It was actually exogenous. And where it actually came from, and it now looks like there's very good evidence to support this, is that this mitochondria that exists in almost every one of our cells, or many of our cells in our body, and they're absolutely critical. These are the powerhouses of our cells. Originally didn't exist there. But where they came from was that mitochondria are endosymbiotic bacteria that were engulfed over 2 billion years ago. So in the common ancestral lineage that gave rise to this group right there, there was a bacterium that was engulfed and basically became part of that organism that gave rise to all the eukaryotic lineage. And we now know that by looking at the sequence, and we can actually see that the sequence of the genome of the mitochondria looks much more similar to bacteria than it does to the rest of the eukaryotic genome. So every one of us is a chimera. Chimera meaning that we have different parts of things. And we've been our ancestral lineage for billions of years, but it's still to this day, we have pieces of DNA in our body that are replicated independently in the nucleus and in the mitochondria. Okay. So let's think about now not unicellular organisms, but multicellularity. What I'm showing you here is a video of a developing zebrafish embryo that was taken by Don Kane a number of years ago. It's another point of pride for the University of Oregon as the university that first used zebrafish as a model organism. And I'm going to show you this video again and just talk to you about what you're actually seeing. So what you're seeing is the embryo moving from a small number of cells to duplicating those cells. This is a process called cleavage. And then all of a sudden, those cells start to look different with respect to one another. And as they do, they start to move down, and you can see these structures start to show up, and you start to see something show up right there and a few things down there. These eventually will become the different parts of the adult body. This is a development that this organism goes through. And this zebrafish is studied because you can easily do this outside the body of the zebrafish and study development. And what's amazing is the development of that body shape early on looks very similar to the development of the formation of the body shape of humans and other organisms that are vertebrates like us. Why is that? Well, this was noticed 125, 150 years ago, and it wasn't clear why. And some hypotheses were that there was just a lot of stability in that formation of the cells and the movement of those cells. But then at some point in the 1970s and 1980s, we started to gather some data, some genomic data that told us maybe it's actually in our genes, in our genomes. And that seems to be largely true, is that when we look at, in this case right here, these are developing zebrafish and human embryos, but even when we look at very distantly related organisms, such as fruit flies and mice that develop as embryos that even the embryos don't look all that much the same. If we look in the genome for example, this cluster of genes right here that don't worry about the little words that are up there, but this cluster of genes are called Hox genes. These are one of the first set of genes that were discovered in the 1970s and really studied in the 1980s that told us that part of the reason that the early development of embryos and the formation of structures and the similarity of different organisms was because there's a similarity in the genes that were actually in the genome. So when we look in the genome, this is just a representation of a small bit of the DNA, and each one of these lines here represents, like we looked at before, different genes. These different genes are actually expressed meaning that remember when we talked about them being transcribed and translated, they do their work up here in the part that will actually become the head of the fly. The corresponding gene in mice, and it ends up that there's a corresponding gene in humans as well too, does its work in the part that will become the head. This part does its work that will become the thorax, and so on and so forth towards the end of the body. So there's conservation, in this case, over nearly 500 million years of the same genes in the same order in the genome in ways that we biologists that is, didn't anticipate at all until genomes were actually sequenced in the 1970s and 1980s. So this was a huge discovery. So that was just a little vignette. I just want to give you one more little vignette. So since that time, there's been a number of different genomes that have actually been sequenced, and this pattern in which the genes seem to be conserved, even over very distantly related organisms like fruit flies and mice and humans, seem to be doing similar functions at similar places in the genome, seems to actually be a trend. So what makes us different? Well, here's the part of that dark matter of the genome that seems to be really important. And for the last few years, a project has been started, and a number of projects, and one of them is called the Encode Project. And they're interested in that other 99% of the genome. That other 99% that doesn't actually get transcribed and translated into proteins like we talked about before. What is that? Well, Encode stands for the Encyclopedia of DNA Elements, and they're interested in understanding and using genomic technologies to say, what are all the functional DNA elements? And not functional by making them being transcribed and translated, but functional just by doing something. They don't make proteins, but they do something else. And when they asked the question that way and used genomic technologies, they found that although 1% of the human genome is coding, making genes, over 70% of the DNA was actually found to be transcribed into that messenger RNA intermediate and is doing something that we really don't know in many cases. We don't know what's going on. 70% of the genome, of our genomes, we don't know what it does. And this is going to be a huge area of growth, is understanding what that part of the genome is doing, especially over the next 20 or 30 years. So any students in the audience, if you're interested in getting into molecular genetics, this is a really interesting area to get into for the next few years. Cool. Okay, so I just want to summarize this sort of overview before we get into the little vignettes for the last little bit of my talk, which is, it looks like that some parts of our genome came from other places. And this is just, the story that I told you about the mitochondria was just one story. There's a number of stories that are emerging where it looks like our genomes have largely been passed down from parents to offspring through generations, but every once in a while there's a new part of a genome that actually comes in from somewhere else. Only a fraction of our genome is actually composed of the genes that are transcribed and translated into proteins. And the rest of that, 99%, we don't know what it does. But what we're starting to understand is that the genes that make the proteins appear to be quite similar, and we can think of them as being building blocks. So what makes us different is not how those building blocks actually look, whether they're bricks or whether they're pieces of concrete, but how those are put together to make, for example, different buildings. So it's not which proteins are actually made, but when, where, and how much those proteins are expressed and how they're actually put together. And that seems to be an emerging theme from genomics, is that it's the manner in which these building blocks are actually put together. So it's a very exciting area. Whew. Okay, so I'm going to just ask very quickly, any quick questions before I move on? Yeah?
You used the term mistake, and I'm assuming you used that loosely.
I did use that very loosely.
Variations.
Yeah. An error in copying, so the DNA itself wasn't copied. But I didn't mean to impute anything into the
outcome could be very
beneficial. It could be, yeah. Good point. Okay. Yeah, I'll take one more question, then I'll.
Is that error in copying what creates new species?
Well, not in and of itself, but eventually the formation of new species has to involve that error in copying. So it's a part of it. It contributes to it, and it's important in the long term, but not in and of itself, only by itself. Good question. Okay, so I've given you sort of a little bit of a background, and I want to tell you some cool stories that are starting to emerge out of genomics studies. And the first one I'm going to tell you is a little bit about how the genomics revolution, or the ability to actually sequence entire genomes of multiple different lineages of organisms, is telling us something about our own past. And to do this, I just want to talk about our most recent or our closest primate relatives. So it's very clear now that our common ancestor, our most recent common ancestor, is one that we share with the lineage that gave rise to chimpanzees and bonobos. It's very clear from DNA sequence data and analyses. And when we look at, for example, the number of chromosome pairs, remember there were 23 in humans. When we look in chimpanzees, there's 24 pairs. And when we look, we can actually see that these 2A and 2B, are very similar to just chromosome 2 in humans. So in terms of the genome organization, in terms of the number of chromosomes, humans and chimpanzees are very similar, and yet we diverge from one another about 6 million years ago. And you can trace this back through time then and see how more distantly related different lineages of primates are with respect to humans. So let's zoom in a little bit and talk about modern human evolution. So at this scale right here. So I'm not going to talk about many of these different lineages of organisms here, but it's been pretty clear for some time. Archaeologists and anthropologists have told us from fossil data that it looked like there were individuals on the planet that their fossils look very similar to humans, but not the same. And we can think that, and it looks as if there were at least several different species of the genus Homo, for example, Homo erectus, Homo neanderthalensis, and our own species, Homo sapiens. And we're going to come back to the Neanderthals in a second. We're going to talk about Homo sapiens first. But there was a hypothesis that was presented a number of years ago that modern Homo sapiens evolved in East Africa, in Kenya, and in Tanzania, and that it subsequently spread out throughout the globe. And that hypothesis had a couple competing hypotheses. with respect to it, and basically it was settled affirmatively as this is what looks like it's happened with modern Homo sapiens, modern humans, evolved about 200,000 years ago, 180 to 200,000 years ago in East Africa, and after that migrated in a couple different directions. One was to South Africa and into Western Africa, but then out through into Europe, Asia, South Pacific, and then eventually into North America. And this is a story that's actually written in our genes. And I'll actually tell you a little bit about the first data that came out using that mitochondrial DNA and told us a little bit about that story that was written in our genes. And this movement out of Africa happened probably about 80 to 100,000 years ago. That's what it looks like, that story that's written in our genes. So even though humans evolved about 180 to 200,000 years ago in East Africa, 80 to 100,000 years ago, modern Homo sapiens moved out throughout the globe. So as I'll show you in a second, they weren't alone. They actually encountered, in particular, Homo neanderthalensis, who had actually evolved and migrated out of Africa much earlier than that. But we're going to come back to that story in a second. But what I want to first tell you about is what the studies of genomes have told us about that ancestral population that gave rise to all modern humans. What they were, where they came from, how they arose. And to do that, we have to go back to the mitochondria and understand that the mitochondria is actually inherited in humans and many mammals in a maternally inherited way, so that mothers pass on the mitochondria in the extranuclear part, outside the nucleus, from mothers to daughters and sons, but only the daughters then pass it on. And I could explain that to you, but one of the best ways to explain that is to actually show you a video from a guy named Adam Cole, who was actually born and raised here in Oregon. And he actually put this together last year because he really likes science. And it was Mother's Day. So this may be loud, so I'm going to start it, and then if I need to, I'm going to turn it down.
To make one me, you just add half of mom and half of dad. That is what I once believed. But now I know that I was wrong. You gave so much to me, Mom, besides one half a set of genes. You gave me nutrients, transcription factors, nearly everything that matters, plus my prenatal environment, transplacental inheritance, mRNA mitochondria that back in the day once belonged to you. I just want to thank you for supplying them. Just like two strands of DNA are spirally entwined, your nature and your nurture inspiringly combined. Scientists remind me, and I find that it is true. Slightly more than half of everything I am is thanks to you. Slightly more than half of everything I am is thanks to you. Mitochondria power the cell, and they have DNA as well. Transcription factors modulate transcription. And since they're in the cytoplasm, the egg's the only one who has them. In sperm, I guess, they don't have the ambition. The sex determination gene means that I'm a guy. You gave me one X chromosome, Dad gave me one Y. X has over 1,000 genes, Y has less than 92. That's why slightly more than half of everything I am is thanks to you. Slightly more than half of everything I am is thanks to you. I roomed in your room for 9 months, and I never paid the rent. Your glucocorticoids helped shape my hypothalamic development. I took your blood and sucked it dry of every nutrient. Gross but true. Sometimes I wonder where the time went. Where did it go? Sometimes I wonder where it all went. I know I'll never understand all you have done for me. I'm not that smart. Since you paid for college, I'll get my BS degree. Bachelor of Science. And I learned it's not BS, but absolutely true. Slightly more than half of everything I am is thanks to you. More than half of everything I am is thanks to you. nearly everything that matters. mRNA, mitochondria, that back in the day once belonged to you. Nutrients, transcription factors, everything that really matters. mRNA, mitochondria, that back in the day once belonged to you. Singing nutrients.
It's not an even 50-50 split. Nearly disproportionate share of the DNA coming from the lung. Really
just like two strands of DNA are spirally entwined. Your nature and your nurture is spirally combined. Scientists remind me, and I find that it is true. Slightly more than half of everything I am is thanks to you. Slightly more than half of everything I am is thanks to you. Slightly more than half of me is thanks to you.
I guarantee that you'll be singing that on the way home tonight. So what does that mean? Well, besides all the other things they were saying were absolutely true, that mitochondria is passed down in a matrilineal fashion from mothers to daughters and so on and so forth. So in this case right here, this is an example of a pedigree, and just represented in blue is the passing down of the mitochondria from mother to daughter, and it gets passed down to sons, but it just then doesn't get passed on any further. And what can happen is by processes that are due to random factors within the population, then the frequency or the number of copies can actually increase such that at some point in the future, all of the individuals that are alive are descended from one individual in the past that actually had that mitochondrial DNA. And when this was discovered in the late 1980s, this was dubbed Mitochondrial Eve. And it was a little confusing at first because people thought what was going on was that there was only one human that was actually on the earth at the time, one female. But of course what that meant was there were multiple females. It's just that all of the DNA was actually derived just from that one female. So all of us in this room can actually look at our mitochondria and look back to that one female that existed probably about 180,000 years ago in East Africa that gave rise to all the DNA that existed in our body. And what's really cool to think about, though, is that what I just said about the mitochondria actually applies to other parts of our genome. It's just that we can actually look at, for example, the Y chromosome in the males, and we can actually point that back to a single male in East Africa. So all of the Y chromosomes can be pointed back to a single individual. And not only that, those other parts of the chromosomes, because if you look at them small enough, they actually don't mix up with one another, they don't recombine, you can actually chase all those back to a single person as well too. So it's very weird to think about, but you can follow those back to different people in East Africa 180 to 200,000 years ago, and you can just imagine looking back through the mist of time and seeing all those people lined up. And there really weren't that many of them, probably 40 or 50,000 of them, and each one of them gave a little bit of their DNA to make up the genomes that all of us have today. It's really wild to think about. So that's one of the stories. Now I want to tell another story very quickly about how when we look back in time and we see all those individuals lined up that gave us the DNA, every once in a while it looks like there's a piece of DNA that goes much further back. We don't actually see that individual. It goes a lot further back. Why does this actually make sense? Well, this is a story that actually has just been unfolding over the last year or so. And this is it's quite exciting. And this is understanding the interactions between Neanderthals and humans. So Neanderthals evolved in Africa and migrated out about 400,000 years ago into much of Central Asia and Europe, and existed there from about 400,000 years ago up to 30,000 years ago, probably even a little bit more recent than that. Not that long ago. So remember I said a second ago that humans actually moved out of Africa, Homo sapiens moved out of Africa about 80,000 years ago. So did they actually overlap? Well, archaeologists and anthropologists have told us for a while that yes, when modern Homo sapiens actually evolved in East Africa and started moving out and spreading into Europe, what they did is they actually, for at least a short period of time, encountered what looked like Homo neanderthalensis or Neanderthal populations. So there were situations in which there were archaeological digs where you could identify Neanderthal populations and human populations. But Neanderthals went extinct, and it's never been clear as to what actually happened. What happened to Neanderthals? Is it that they were just extirpated? Did modern Homo sapiens actually interbreed with them? And to answer that question, a project was started a number of years ago by a number of people, and it's really been pushed forward by this gentleman here, Svante Pabo. And what they said is, well, let's just look in the genome. Let's look at Neanderthal genomes. And this seems kind of crazy because there aren't any Neanderthals around, but what they decided to do was to go in and actually dig up Neanderthal fossils and said, well, there's a possibility, but we might actually find DNA in there. And lo and behold, they did. Now that DNA isn't as intact and in huge chromosomes like we have in our bodies because it's been in the ground for tens of thousands of years. In this case right here, they've actually were digging up fossils that were probably 45,000 years old. So there was DNA in there, but it was highly degraded, meaning it was broken up into little chunks. And what they needed to do is work in these pretty amazing conditions where one of the things that they wanted to do was not contaminate the DNA with their own DNA. So they were working in these caves in these situations and back in the laboratory in which they were trying to cover up every aspect of the body so they didn't contaminate it with DNA. So to go through and actually sequence this basically had to wait for a revolution in genomics. And this revolution occurred in 2006, early 2007, with the development of new sequencing technologies that are called massively parallel sequencing. Previously, sequencing was done just one bit of the genome at a time, one gene at a time. And what happened was a number of engineers got together and said, hey, what if we actually go through and build a machine that's basically the size of a Coke machine and put very high performance optics in it, along with high performance microfluidics that will allow us to go through and flow through DNA on something the size of a microscope slide. But then instead of just sequencing one piece of DNA at a time, let's sequence millions of fragments of DNA at a time. And when they did that, they actually could go through and flow it through and take pictures over and over again. And what this allowed biologists to do is instead of sequencing just a few hundred or a few thousand base pairs at a time, now 20 to 100 billion base pairs can be sequenced by this machine each week. And the University of Oregon has one of these machines, and we do a lot of sequencing every week. So whereas the Human Genome Project that I showed you that was finished in 2000 cost about $20 million, probably a lot more than that, to finish one human genome, right now, for just a few tens of thousands of dollars, 5 to 10 human genomes can be produced each week. It's amazing to think about. So what that allowed them to do is take all those fragments of DNA and sequence them simultaneously, and it was a big amount of work, and put them back together. And so what did they actually find out? So what they actually found out was that when they looked at a number of different sites in which they actually isolated Neanderthal DNA, and they did it through in Spain and in other regions throughout the Central Asia and around the Mediterranean, and then looked first at the mitochondrial DNA. Well, just like we'd expect, the mitochondrial DNA of all modern humans coalesces or moves back to a point right here that's a single ancestor, Mitochondrial Eve, before the common ancestor of the Neanderthals. So that first evidence from the mitochondrial DNA seemed to suggest or indicated that, well, there might not have been any inbreeding going on because all of the genome, at least the mitochondrial genome, traces back to a single person, a single mitochondrial Eve in Homo sapiens before it does back to a common individual with Neanderthals. Once the genetic information from the rest of the nuclear genome started coming out, what biologists realized is that there's a little more interesting things going on here. One of the first big surprises came out about 2 years ago, in which there was a study of a gene that actually makes this protein that goes across membranes, and this protein is called an MC1R gene. It's a receptor that actually has variants to it that is important in making us either have dark hair or red hair. And it ended up that, just like in modern humans, there's variants for this gene, different alleles. There were actually variants found in Neanderthals. So this is a reconstruction of what maybe a Neanderthal child looked like with a variant of the MC1R gene that actually made it redheaded. So instead of those dark beasts that we showed you at the beginning, that maybe Neanderthals actually didn't look that different from humans. And the most recent information, this is the most exciting, is that the first draft of the Neanderthal genome was produced just about 3/4 of a year ago. And that Neanderthal genome is about the same size as the human genome, about 3 billion base pairs in length. And when you look through the Neanderthal genome and compare it to modern human genomes, you can see that it's quite divergent and different through most of the genome. However, there are little bits of the genome in human populations that look very similar to the Neanderthal genome, small regions, but only in certain human populations. And it's all in modern Homo sapiens populations. The only modern Homo sapien populations that have those little Neanderthal genomes are the ones that were originally derived from humans that actually were part of the diaspora out of Africa through Mediterranean and out in the rest of the world, not found in the African populations, only in those humans that actually moved out through the Mediterranean. So only in those regions in which modern Homo sapiens actually interacted with Neanderthals was there an indication that there was potentially interbreeding going on. So this is, I would take this with a grain of salt. It's the newest genetic data. There's genomic data. There's other interpretations, but it changed my mind. I didn't expect it until I read this paper, and I thought, this is a little convincing that there might actually have been some of our genomes that exist in humans that actually migrated out of Africa ended up being movements into individuals, for example, Chinese and French, but not into different populations of African humans, Homo sapiens. Again, another surprising discovery that's come out of Genomics, and this was published just 3/4 of a year ago. Pretty exciting stuff. So, just to summarize this part, and then I'll just finish up, move to my conclusions here in a second. We're changing the way we're thinking about our past because our genomes, we now know that mostly of our genomes are in our nucleus, but at least part of our genomes are in our mitochondria. And that mitochondria originally was pulled into our body, or it was sequestered billions of years ago, and has stayed with us ever since. Aspects of the mitochondrial genome and the nuclear genome actually move back and forth. We can actually tell that. And that mitochondrial genome tells us something about our recent history so that we can actually think about and look at all of the tracing back of that mitochondrial genome and other parts of the genome to a small group of modern Homo sapiens 180,000 to 200,000 years ago. But not all of our genome. At least some of our genome actually seems to be transferred over from some of our closest relatives that are not Homo sapiens, Homo neanderthalensis. Very cool. So what I want to do very quickly is just tell you one last quick story and then finish up, which is another way that genomics is actually telling us something about ourselves very differently than it was before. And this is using an approach called metagenomics. So what does that mean? So before genomics, we were talking about genomes of an individual species or different human genomes. What a metagenome is a metagenome is taking all of the individual different organisms that actually exist in an area and then collecting those together and sequencing the genomes to try to tell us something about the abundance and distributions of organisms that are actually there. So why does that actually matter? Well, this is telling us all kinds of stuff in many different ways, but a lot of work's actually going on in this area right now doing metagenomics and looking at communities or groups of organisms that are these microbes that I told you about before, bacteria and archaea. And this is happening in a variety of different studies in a variety of different ways, but one way that this is actually happening is in the context of humans themselves. So there's a project that's been started called the Human Microbiome Project. What does microbiome mean? Well, it's that community of microbes, bacteria and archaea, that actually exist on the human body. So for years we've known that bacteria actually are on, in, and around us, but most of the time we thought about it as something that caused diseases or caused problems for us. But what's becoming increasingly clear is that when we look at those bacteria on our body, and especially if we look at different places, for example, if you take bacteria from the mouth or different parts of the skin, you can actually find that if you take those bacteria out and use that sequencing approach that I told you about before with the next generation sequencing, the massively parallel, you can find all kinds of diversity such that even from your front teeth to your back teeth, you have different bacteria that exist there. From your outside of your skin on your elbow to the inside of your elbow, different bacteria. And many of these bacteria seem to be actually living in a commensal or even in a symbiotic way so that they're not harmful, they're actually very critical. So this Human Microbiome Project, for example, there was a paper published just 6 months ago where they went through and did a massive amount of sequencing from microbes that were collected from all kinds of parts of the body. And what they found was that when we do this, if you just make a big tree, just like we did before, but all these species, even just looking on humans, 178 new species that had never been identified before on our bodies. nearly 30,000 new proteins that we had never known about before being made on or in our bodies. Very cool work. And just to finish up, I'll just make a highlight here that, so what are these bacteria doing? Well, it's kind of hard to tell what they're actually doing in our bodies, but it's clear that they might be playing roles that are really important. So in some cases when we, for example, have bacteria communities that are different than they should be in our guts, diseases are actually associated with them. Are they causative or is that a consequence? Well, one way to get at this is actually to move to, again, our friend the zebrafish. And I just wanted to point out some work that's being done by Karen Gilliman. She's a faculty member in the Institute of Molecular Biology here at the University of Oregon, studying zebrafish to ask that very question. So what she's doing is she's actually taking zebrafish and using a whole bunch of cool approaches where she can make them transgenic or put different aspects of the genome in there. And specifically, she can also make them axenic, or she can get rid of all their microbes. There's a cross section of a gut of a zebrafish with microbes inside, get rid of all the microbes and rear them, and then reintroduce them into the inside. And you might ask, well, what does it matter what the microbes are in their gut? It ends up that it matters immensely. What she's actually finding is that the microbiota inside the gut are important for intestinal differentiation. So the microbiota are actually the ones that are making signals that are important for which cells become proper intestinal cells. So without the correct microbiota, that development doesn't happen correctly. The rate at which the epithelial cells, or the cells that actually line the gut, expand is important based upon the microbiota. And even more fascinating to me, the first time I heard about this, is the proper maturation of the immune system doesn't occur without the correct microbiota. So if we take all of this together, we can actually think of ourselves now in the present as expanding not only to be only our cells in our body, but actually the order of magnitude more cells that we have in and on our bodies that are actually microbial cells. So when we think about ourselves as an individual, we've got all these cells in our body, but we also have this community of cells on us. And that community is very much a part of our cells and is very important for things like the maturation of our immune system, the proper oral health, things like that. And this is something that's really come out of the metagenomics work or sequencing, such that you can think about this in terms of both the genomes of the microbes and the environment, whether you're exposed in the early infancy, prenatal exposure, or exposed to antibiotics and urbanization, and how those things actually lead to a healthy community or an unhealthy community can actually affect the individual that we are, whether we have, for example, gut diseases or not. So that's another case where genomes are being extremely important in terms of the way we think of ourselves as not just single organisms, but community of organisms that exist together. Okay, so I think what I'm going to do is skip to my last little bit there, which is I had one more little bit that I was telling you about some work that a researcher in the University of Oregon and I, Wei Zong, are doing about understanding how cancer progression and sequencing different cancer cells and what that might tell us. We haven't done the work yet, so I'll just tell you about it next year or 2 years from now. But one of the points that I want to finish up with is that, ready or not, this genomic information is coming. It's coming right at us. And so what I decided to do is I Googled today, speeding train. And what I actually found was this one first. And I thought, well, that's actually pretty appropriate. So the information is going to be coming at us. And this is not just a scientific issue. It's actually an ethical and a sociological issue, which is that what do we do with it? Well, if we actually use the information correctly and in ways that fit into our morals, our ethics, and our society, we'll be cruising along nicely. If not, it is going to be a train wreck. And Lee Silver will really talk about this quite a bit. And I'll just give you one example. So the doctor will see your genome now. So I decided to put up here, those of you who actually know Doctor Who, it's a show from BBC. There we go. Somebody's a very excited Doctor Who fan. So Doctor Who is he's an interesting guy because he actually can't die. He has to regenerate himself over and over again. It's actually great for the show because it's like the show doesn't seem to die. It's been around for 35 years. And this is one of the first Doctor Who's. This is a later Doctor Who. But basically, he can travel through time and see the future. And this is one of my favorite characters, the Ood, that are actually can sort of look into the future, but imperfectly. And that really is what's going to be happen for us, which is that we'll be able to look into our futures in ways that we never could before, but imperfectly. And we really have to get a handle on that, which is that in 2003, the human genome cost $20 million to sequence. Right now, I've seen estimates of $20,000 to produce a human genome. Any one of you, $20,000, you could sequence all 3 billion base pairs. 2015 projections of 2,000. By the time you get at that point, it's very realistic that you will go into your doctor and will be asked, would you like a genome scan? Would you like your entire genome sequenced? What do you answer? Well, yeah, I want that information. No, I don't want to know at all what's going to happen. And well, there's some benefits to this, of the genome revolution. One is that we might actually be able to tailor treatments, for example, to different individuals. We might actually be able to say, here's your genome sequence. You have these different risk factors, but we also know that this drug will work for you, but this won't. Hey, that seems like a pretty good thing. Well, how about growing new tissues? This is not something that's sci-fi. This is actually happening right now. These are researchers at Georgia Tech that are actually growing skin cells. Research over the last couple of years has been able to figure out ways to take cells that were totally differentiated and push them back to an undifferentiated state where they can actually become subsequent cells. And it actually involves fewer genes than we thought. As a matter of fact, only 4 genes. So we can take it back to that state and actually make new tissues. So having complete genomic information might assist in the ability to, for example, grow new cartilages for your knee if you have a bad knee like I do. But there's some real serious perils here as well, too. So how should we actually use our own genomic information? So should we actually make decisions about our own treatment? Make sense? How about our own future? Yeah, planning for the future. How about decisions on who to marry and who to mate and which children to actually keep? We already do some amniotic testing. Sorry, didn't mean children. Decisions to actually make about reproductive decisions. But also, how should this information be kept safe? The last email that I got before I actually came here was from one of my credit card companies saying, we're sorry, some of your information was inadvertently released. It's not a problem. And so that was my last email before I came here. So that's something that we really should consider. Scientific literacy is going to be extremely important in this. So we all have to know, for example, like Bryn set up at the beginning, that genomics is not destiny. Genetics or genomics is not destiny, and what probabilities mean. And when we say there's a certain probability of getting disease, certain environmental influence, what does that actually mean? How do we make those decisions? But the answers to these questions that we laid out here are not just scientific. They actually involve ethical and moral questions as well, too. And that's what this series is all about, is to try to get people to actually think about what this genomic revolution means, but more so, what this genomic revolution will mean in the near future when all of us have this information in our hand. What are we actually going to do with it? And I'm really looking forward to the next couple talks in the series. Thank you for listening, and I'm happy to take questions. Bryn, do you want to give me 3 minutes to funnel out? Okay.
So, I don't understand why this doesn't trace back to a single individual ancestor. Is that just serendipitous and happened that way? Because, as I understand it, there was a point when there were maybe 2,000 of us. Right. We all, and presumably, all of those reproduce, all of the women reproduce. Why are we not seeing why are we not seeing 5 individuals or 10 individuals?
That's right. Yeah, so it actually ends up that it could be a variety of, the question was, so what is the logic behind the fact that, for example, right now, we can trace all of our mitochondria back to a single ancestor? What process would actually lead to that? Well, there's a couple processes. One is natural selection itself. If for some reason that genomic region actually gave some benefit to the individuals that carried it, it would increase in frequency in the population. And that can actually happen quite quickly. But even in the absence of natural selection, there's another process that can occur in populations called genetic drift. And it's actually just it's a consequence of random sampling of alleles from one generation to another. Even though every female is reproducing, just some females will produce more, some will produce less, and that's not tied to that genomic region, but just that process of having some alleles that are passed on from one generation to another, or even just being put into one gamete or the other. That random process of sampling from generation to generation can actually lead to an increase in the frequency and what's called fixation of that over time.
So one of those two explanations, one of those two reasons could explain the fact that it could have happened that we would see the results of maybe 2 or 3 or 4 individuals or 10 or 100, but it didn't happen that way.
Yeah, it didn't happen that way for mitochondria, but your point is a good one, which ends up that point to which all of the regions of the genome coalesce back to is variable. So some of these coalesce back more distant in the past and some more recent. And it's because of the random sampling process that occurs called genetic drift.
Could I just add one plug to everybody to remember to have your children play and eat in the dirt and beware of the antibacterial microbial soaps.
Thank you, and thank you for your talk. It was fascinating. From what little reading I've done about all of this, I understand that doing the genetic mapping turned out to be far less complex than what they're turning up now with epigenetics. And I'm wondering if with epigenetics and the methyl markers that seem to signal what's going to happen then genetically, if you could talk about that a little bit. And then the second question is, can you talk a little bit about how viruses may interplay with what kind of DNA information the bacteria are carrying, and then what gets turned on in the body for how it's going to work, and how that might affect the methyl markers and epigenetics.
Okay. So I'll start with the epigenetics. So as you can tell from these questions already, excellent ones. There's so much information, obviously couldn't cover it tonight. Even in the next few lectures, even 20 lectures, we couldn't cover all of it. One important point is that not all of the information that gets passed from parents to offspring has to do with changes in the DNA sequence. They can also be changes in tags that actually happen on that DNA sequence itself. Not a change in the base pair, but a change in the tag. And the point that he was making, that's called epigenetics. So it's inherited from parents to offspring, and it might be change in a group that's actually a part of the molecule that's on the DNA. So that actually can be important for a small number of generations, but eventually those methylation patterns will, or those epigenetic patterns will change over time. So they have important consequences for the short term. When we ask why is an offspring similar to a parent, part of it's due to the alleles, the different gene variants that are inherited from the parent to the offspring. Part of it's actually due to these epigenetic factors. And that's a huge area of research right now. We know that they're important, but I don't think that most biologists would say we have a good handle on exactly how they're important or what they do. That was the first question. The second question was about viruses and how those are important for bacteria. So there's, just like viruses can attack us, viruses called bacteriophages attack bacteria. And in many cases, those bacteria that are attacked will actually just be killed. But in some cases, depending upon the virus, and same thing with us, they can actually be, if it's a certain type of virus, retrotranscribed into the DNA of the bacteria and into our cells. And in doing so, they can actually induce mutations, for example, if they're retrotranscribed into a gene. And then it'll actually be a mutation in the gene. And also what can happen sometimes is that process of retrotranscription can be a little sloppy, so it can actually, the virus itself can carry bits of RNA that get retrotranscribed into DNA that then get passed from one organism to another. I know that happens, but I have to say I'm not an expert on the frequency or the importance of it, so yeah. Steve.
Okay, hi. At several points throughout your talk, particularly about hominid evolutionary history and trying to get some of the timing there, particularly if we're trying to figure out if Homo sapiens and Neanderthals might have coexisted at the same time. There are a lot of dates that are getting thrown around in there. So how can we look at genetic variability today and tell something about when events happened in the past based on that?
Yeah, that's a good point. So I didn't actually go into another thing that I didn't go into. So how do we actually know when we look at a set of DNA? All we have to go on is a sequence of organisms that are alive today, the ones that are extant, or in some rare cases, organisms for which they were they were fossils but had some DNA in them. How do we actually date these things? Well, it's actually pretty hard. It's what's called the molecular clock. If we assume that molecules change at a certain rate and substitute at a certain rate, we can actually then ask, if we look over a set of organisms, that they're this different, and if we knew that those molecules evolved at a certain rate, this many differences must have accumulated over this many years. Simple, right? The problem is that different molecules evolve at different rates, and largely it's due to the mutation rate. So different parts of our genomes evolve at different rates. So when we look at the mitochondria, for example, the mitochondria has a higher mutation rate and evolves at a faster rate than many other parts of our genome. And what we need to do is, and what molecular geneticists try to do, is date and say, well, we know that this split, for example, between Neanderthals and humans happened at least this long ago, because that's the first time we see both Homo sapiens and Neanderthal fossils, for example. Or another scenario in which you can positively say that, for example, Darwin's finches on the Galapagos Islands, that this species arose when this island was formed, and this species arose when this island was formed, and geologically we know when those islands were formed. You can calibrate the clock and say, for these molecules, we know at this time point, this was this long ago, this is this much divergence, we've calibrated it, let's look at another organism and then say, this calibrated clock, that's a molecular clock. So that calibration process, though, is very sloppy. So the dates that I was giving you and throwing you around, if you actually look at those, there's plus or minuses on either side of those. So that's another. You shouldn't only take those with a grain of salt. Patrick.
Yes, Dr. Cresko, I was wondering if you could comment on whether or not there's any evidence for relationship between the Neanderthal genome and the political inclinations.
No comment.
My question has to do with the symbiotic relationship between us and our gut flora,
Yeah.
if you will. Does that mean that we're no longer a single organism? Because we know if that's gone through too much antibiotics, what happens to us? And they all say, go eat some yogurt or whatever, and will the doctors of the future be prescribing bacteria for our innards, much the way they prescribe other things for the rest of our bodies?
Yeah, I'm actually going to ask you a question back, if you don't mind.
Yeah.
What's a single organism?
I don't know, but I guess I'm thinking that we are perhaps not one unit, whatever, anymore, that we're reliant on these other creatures.
I didn't mean to put you on the spot.
No,
The
that's
reason I said
okay.
that is
I'm
because
confused too, so.
No, it's not. That's a question that we actually ask each other as biologists. And when you say an organism, what do we actually mean by that? And part of the reason that I brought up the mitochondrial DNA part was that, well, sometimes we say an organism is a single physical unit that actually has a genome in it that was ancestral descendant inheritance. And that makes sense, except the mitochondria has been with us for a long time, and it's passed from parents to offspring. But at one point, it was actually pulled in. So does that mean that we actually had two organisms that became one? Yeah, that makes sense. And so in that case, it's kind of clear because the mitochondria is always passed from mother to daughter, and so on and so forth. And in the case of the gut microbiota, and I'm going to go out on, this is not my area of expertise. There's somebody in the audience who's several people in the audience who are much better at this. What if it ended up that the microbiota were passed from mother to daughter in a similar fashion, though, so that perhaps the birth canal was supplying the bacteria that then were passed on to the daughters and sons as they were born? In that case, you'd have this regular inheritance of the bacteria from the gut of the mother into the gut of the children in the same fashion as the mitochondria are inherited. It gets really kind of loose as to say then it's a single organism with different a single human organism, but then the microbiota is different as compared to almost like a meta-organism. So,
Can those microorganisms get along without us? Or have they evolved in a way that they are so dependent on us that they...
all right, I'm going to kick it out to the experts in the audience. Brendan, do we know about most of the gut microbiota? Can they survive outside of the gut environment?
So of those that we know, there certainly are examples of ones that are rare to find outside humans, and those that seem to be commonly found. So the answer is unfortunately a qualified maybe.
But,
examples of both, absolutely.
yeah, but I would encourage you to keep asking these questions, and I'm glad that you asked that, because this is one of the questions that I think genomics and metagenomics is actually making us think about is, who are we as an individual? We're our cells, but our bacteria are our cells too. What makes us, and those bacterial cells, in many ways, made us the individuals we are, adult individuals that we are today, because of our immune system, for example, being adaptively formed. But maybe there's other traits as well too that are important because of that. So, good question. Yes, go ahead. Hi.
How is studying genomics helping us to understand speciation?
Good question. Another huge area. So I'm going to give you a very short answer to this, is that there's been models for how new species actually form in terms of genetic changes for a number of years. And those are clearly occurring, in which you actually have a single lineage that actually goes into two different environments, and different genes that make different proteins start to change a little bit. And when they come back together, they don't play very well with one another. So at that level, those genes that actually, they don't play very well together, they actually will end up killing the hybrid organisms if they're actually formed, if you end up having what are called incipient species come back together. So at the level of genomics, it doesn't matter, you can actually look at it at the level of the gene. But it's clear that in other cases, genome changes actually matter, especially if there are changes in the size and shape and position of the chromosomes themselves. Because what happens when the organisms that are starting to diverge come back together, if their genomes are different enough, they can actually make offspring that might be completely healthy in terms of being energetic and viable. And a good example of this is mules, so crosses between horses and donkey. But when they actually go to make their own gametes, because the process by which the replication of the DNA material and the sorting of it into the gametes happens, it can end up being problems in terms of the formation of the gametes, because different combinations of the genetic material will go into each gamete, and in most cases that's deleterious or downright, or usually lethal. So genomics seems to play a big role, can play a big role if there's divergence enough in the size and shape of chromosomes, so that when they come back together in a hybrid, then subsequent offspring can't actually be made. So that's recent work on that seems to point to that pretty clearly. You're welcome. Yeah.
Assuming that we know that about 85,000 years ago there was a mitochondrial Eve, and we do know that there are some rogue DNAs in that mitochondrial Eve, or that are present today, do we have any idea how old those rogue DNAs are? Do they possibly trace back to Neanderthals and even possibly further back?
Yeah, so I missed the one, you said rogue DNA?
Well, just the unusual ones that
I
are
see
outside of the normal scope that we know as being from the mitochondrial Eve.
Right, so that single mitochondrial Eve may have actually given rise to, she clearly gave rise to the mitochondria that was passed on to her daughters and her granddaughters, that then for one reason or another increased in frequencies and became in all of us. But it's an open question as to whether, it's not unknown whether other parts of her genome are actually present in us at all. Maybe not. So it may be that all the other parts of her genome actually died off. But there might be, for example, another part of human chromosome 1 that's small that actually was passed on from another human that was alive, not exactly that same time, but maybe a little bit younger, a little bit older than that mitochondrial Eve. But at least some of those individuals that actually interbred with Neanderthals then passed on what looked like Neanderthal genomic regions that became that are seen now in modern human populations. So I think your question though is there DNA that seemed to be an outlier from about the same time at which mitochondrial Eve might have existed? And the answer to that is also yes too. There actually does seem to be cases where there's DNA that seems to be older, but that one's a little more shaky.